Hello, welcome to the final video for cellular respiration. Yes, we are here and we are now on the last part. So we were done with glycolysis, we also talked about fermentation, and then if oxygen is present, it will continue to pyruvate oxidation after glycolysis, and after that it's Krebs cycle. And so finally, today we will be having the electron transport chain. Okay, so let's see what is the fuss in electron transport chain. Okay, so electron transfer chain or ETC and oxidative phosphorylation. So the location of this process is in the inner mitochondrial membrane. Okay, and it uses ETC. So we have different proteins that help us do the job, and ATP synthase, the enzyme, to make ATP. So overall, what happens here is that the electron transport chain pumps our hydrogen ions, they are protons, across inner membrane, which then lowers pH in inner membrane space. So I'll show you what it looks like. Okay, so this one here, this big blue circle represents our mitochondria. And inside, let us take a look. So this is the matrix. Again, it's the innermost part of our mitochondria. And the Krebs cycle is happening over here, right? So we are creating a lot of products as well. So it is said that NADH and FADH, both electron carriers, will help us to start our oxidative phosphorylation. So let us look at NADH first. So remember that NADH is an electron carrier, right? So it will be oxidized, means that it will lose an electron. So that electron will now travel into different electron carriers. This one, the blue circles that you find. So it will travel there, and once it's doing that, hydrogen ions will exit the matrix. So it will go to the intermembrane space. They will go there. So notice we have high hydrogen concentration in this part. Okay, so they're going there, and the electron continues to travel. And finally, the last electron acceptor that we have is oxygen. Okay, so if you remember this one right here, it's similar. It's kind of similar with photosynthesis, right? So, but here, oxygen is needed because oxygen will accept that electron. Oxygen is the final electron acceptor. But in photosynthesis, it's opposite because oxygen is produced. Okay, but anyway, because of that, so oxygen accepts an electron and it will now result to the product, which is H2O or water. Okay, and then next, so that's the case for NADH. Then after, let's look at FADH here. So actually, it's still the same, but if you compare our proteins here, see, 1, 2. Okay, so we are using fewer proteins. So in FADH, it will still be oxidized. So that electron will travel through electron carriers. And then hydrogen ions will exit. Still, they will go to the intermembrane space. And then the final electron acceptor is our oxygen. And also, it will be combined with with 2 hydrogen ions, which will result to the creation of water. Okay? And then, so because of that, so notice the thing that is happening here is that hydrogen ions are going to the intermembrane space. So this is now an area of low pH and high hydrogen concentration. So because of that, the hydrogen ions will, as you follow the arrow, so it will enter through the ATP synthase, the enzyme, this red one. So the enzyme will do its job and powers the creation of ATP. So again, it's phosphorylation where we add a phosphate group to ADP. So it will result to ATP. Okay? So once again, just to summarize everything, NADH and FADH, they are needed so they can give their electrons, and then that electron will travel and hydrogen ions will exit, and then it will enter the ATP synthase to make our ATP. So because of that, a gradient is made. Okay. Alright. So also, let's look at this part. So the hydrogen ions then move via diffusion or protein motive force through ATP synthase to make ATP. That's what we saw in the picture. And all NADH and FADH converted to ATP during this stage of cellular respiration. And here, okay, take note of this. Each NADH converts to 3 ATP, while each FADH converts to 2 ATP. So it enters the electron transport chain at a lower level. So fewer ATP are made by FADH, but just by one molecule. So it's still okay. Alright, so if we look at our chart here, we are all done. So again, for cellular respiration, the first part is glycolysis, where we transform glucose to be converted to pyruvate. And then if oxygen is not present, fermentation will occur, an anaerobic process. But if oxygen is present, after converting glucose to pyruvate, pyruvate oxidation will take place, where pyruvate will be converted into acetyl-CoA. Then after, it will also have a migration from the cytosol to the mitochondria. And then afterwards, Krebs cycle will take place. This is now inside in the matrix. Okay, so the final product here is we have citrate, right? So acetyl-CoA will bond with OAA. So we have different products there as well. The yield that we have. And then lastly, we have the electron transport chain. Okay, so let's sum it up here. Let's look at the maximum ATP yield for cellular respiration in eukaryotes, since if you think of it, the point really here is we have to make some energy. So cells can continue to do their job. So organisms can still continue to live. Okay, so let's see how we make ATP. Okay, so this is again in eukaryotes. Okay, plants, cells that have nucleus. Okay, so here, glucose will undergo glycolysis. This is the first step. So glycolysis will give us different products. We have 2-pyruvate. We have 2-NADH, which will go straight to our oxidative phosphorylation. And we have our ATP here, so it will be included in the total count. And then in the pyruvate, so it will be converted. So this is now the grooming phase. Pyruvate into 2-acetyl-CoA. Then we also have NADH. Okay, and then after that, Krebs cycle will happen. Okay, so there is the creation of ATP, FADH, and NADH, and this will go now to ETC and oxidative phosphorylation to create 18 ATP. So all in all, if we add all the ATPs made, that gives us 36 ATP maximum per glucose. So one glucose molecule creates about 36 ATP for eukaryotes. Okay, let's look at the total yield over here where we add all the ATPs. So that's 36, right? Okay, so there are other cells as well aside from eukaryotes. So we have prokaryotes like bacteria. So they are also organisms that are capable of cellular respiration. So this is their total ATP yield. And notice this part. Okay, look at the total ATP. So per molecule of glucose, they have 38 ATP in total. So look at this part over here in black. So 6 ATP will be converted from 2 NADH in the glycolysis part. Okay. So if you look at it, it's different from prokaryotes and eukaryotes, right? So what makes it different? The answer is because eukaryotes have membranes. So they have to go through different membranes, and going through membranes requires energy. That is why in eukaryotes they have fewer total ATP. So 36 ATP here and 38 ATP in prokaryotes. Okay. In addition to glucose, what other various food molecules are used in cellular respiration? So, I have started this video lecture by showing you how glucose is split, right? So is that the only thing that is broken down? Is glucose the only thing that our body needs to create energy? The answer is no, because there are other food sources, right? Other food molecules, and those are carbohydrates like polysaccharides, fats, glycerol and fatty acids, and proteins. So they're monomers amino acid. Okay, so all of these other organic molecules can be used for fuel. That is why we can also have carbs, fats, and proteins in our diet, and we can still have energy from them. Okay, so yeah, I guess that is it for this video lecture. Basically, we were able to learn about how organisms break down their food so they can obtain energy for survival. I hope you learned a lot today, and if you have a lot of questions or clarifications about this topic, you can watch the video again. We have different parts. It's divided into subtopics, so you can easily search for whatever you want, whatever topic you want, and you can also ask your teachers about this. It's Ma'am Florence, and congratulations on making it this far. Thank you!